Would it be resistant to this factor or not? So also, combined with the feature that cancer therapy is not applied with the same therapeutic rate to every type of cancer, means that the genetic profile interferes and the microenvironment interferes with the behavior of cancer cell treatment. So it's essential to know, if it's possible, from the beginning, how this cell or how this type of cancer will respond to each kind of therapy. So already we know that empirical lines of therapy respond to a specific rate. For example, we know that if we apply a FOLFIRI in a colon cancer as second or third line of therapy, we may have a response from 15 to 25%. So what happens with the rest 75%? Why they do not respond? We're talking about the same type of cancer microscopically. In microscopic world, it's a completely different story. The older methods since the beginning was the clonogenic assays, which we took the primary tumor out by surgically remove, and we put them mainly in soft solid culture media, and we expect to produce explants, and those explants produce colonies, and then we put the cytotoxic drug up on a colony and see if we have a decrease or not of those colonies, which means long period of exposure, which means that we grow a clone from the total population, and doesn't mean that there is a linear relation between the outcome of the culture and inside a patient. Then we developed the colorimetric assays, simple assays, still used, very simple, with trypan blue 0.4%, which is actually dyes, stains, which normally they do not penetrate when the membrane of a cell is intact. So when a cell destroyed, become dead, so we have cell membrane perforation and the dye goes in. So we can count how many cells they have perforation in the membrane and see how many cells are dead under treatment and how many cells survive. A spectrometric assays, which is still used in pharma industry and in research, but it's far, far away from clinical practice, like crystal violet, sulfurodamine, or methyltetrazolium. Both kind of tests, the crystal violet and SRB, they stain the protein inside the cells. So when we have a dead cell, there's no protein inside, or the protein level decreased. Or the MTT is a substrate of mitochondria, and it forms formazine crystals inside. And then you can count the optical density in both methods, and you will see by the optical density the cell population if you have primarily perform a control diagram. The most latest of the simple assays was the luminescence assay by luciferin luciferase. Luciferase is an enzyme who transforms luciferin and helps them to produce photons. But this is according to the metabolic activity of the cell. So we have a cell kill, cell destroyed by under cytotoxic effect. Then we do not have activity of luciferase, so there is no photon. This is bioluminescence. What are the disadvantages of all those assays? They are not linear, which means that the number of cells and the signal or the measurement is not linear, which means that if we have an increase or decrease of the signal, you may not necessarily means that you have a change of cell numbers, that they are viable. Few of them are reproducible. They provide reproducible results. For example, MTT has big standard deviation, very popular in journals. But if you put them in raw data, you would be surprised about the deviation of the results. They need long period of development for cancer cells ex vivo. Clonogenic assays means that we need to spend 4 to 6 weeks only for cell growing, which means that you spend valuable time, needs long period of time to develop measurable colonies for evaluation. This is for clonogenic. And most of all of those tests needs tissue to perform. As you know, when we have a tumor, only a small proportion of this tumor is cancer. A tumor is composed by cancer mass and the stroma cells. The cancer mass composed by the subpopulation of cancer cells, and the stroma cells are macrophages, lymphocytes, endothelial cells that they support the existence and the survival of the tumor. So when you remove a tissue and you perform tests, you perform tests from those cells as well. So it means noise. Noise means that your result is not become clear from your cancer cells. That's why we need to proceed further for novel techniques, which we need accuracy, sensitivity, specificity, as many information as we can concerning